So the next speaker is Nicolas Rascovan. It's just 3 weeks that he started his new lab in Pasteur, and he's going to tell us about a microbial perspective to the human past.
Right, thanks. Well, first of all, thank you for giving me this opportunity. It's really an honor to be giving a talk here. So as Luis said, it's only been 3 weeks that I am a PI of a new lab in Pasteur Institute. So I will be talking a little bit about the work that brought me here. I don't have much to show about what the lab has been done, because in 3 weeks we didn't do much. So the title of the talk is A Microbial Perspective to the Human Past. And in particular, the lab will be interested in using the perspective of time to better understand the present, in particular the perspective of time to understand microbes and microbiomes. And a quote that has been always been very enlightening for me was this from Tadeusz Dowshansky, which says that nothing in biology makes sense except in the light of evolution. And I also like to add to this quote that the historical perspective also helps us to better understand the past and the present. So in this talk, I will be inviting you to make a time travel, and our time travel starts only 335 years ago, when these two guys did a revolutionary discovery. Because what they discovered, essentially, is that we are not alone. They discovered that life, as we know it, life as we can see it with the naked eye, is just a part of biology, a part of life, and there is a whole universe of microorganisms that live around us, within us, on top of us, everywhere else. A second revolutionary moment in the history towards microbes is around 160 years ago, when Louis Pasteur and other people in their time, they did the discovery that actually these microorganisms were responsible of many of the infectious diseases that so far, until that point, we couldn't really explain. What the counterside of this major advancement in human history and towards medicine is that ever since we got an idea that microbes are bad. The microbes are pathogen, the microbes has been associated ever since with infectious diseases, and phrases like that we are in an arms race against microbes has been used ever since. But in fact, over the 20th century, we have started to see that actually there are many more microbes that we can see in the microscope that we can actually culture. And so far, during the 20th century, the only way that we had to really study microbes were by culturing them and by doing experiments on them. So only 35 years ago, there was this concept of the Great Plate Count anomaly that says that there is only a tiny fraction of the microbes that we can see that can be actually cultured and therefore studied. And only 30 years ago, some people, with the possibility to sequence DNA sequences, we discovered that actually we could amplify the 16S ribosomal RNA genes from microbes, bacteria, and archaea, and they started to discover that the diversity of microbes, the tree of life of microbes, were much larger than those that we could cultivate in the lab. And only 15 years ago, so yesterday, we started. There was the high throughput sequencing technologies were introduced, and actually, thanks to that, we started to discover that the tree of life is much more larger than we thought, and actually that most of the branches in the tree of life are microbial. And eukaryotes are only here, in a very tip of the tree of life. So one of the implications, of the many implications of this discovery, is that we humans actually can be seen now as very complex and multicellular biological entities, because in fact we are populated on the surface, inside us, around us, by a huge diversity of microbes that cannot be cultured and therefore cannot be studied by simply cultivating these microbes. And this brings us to another time travel through the whole history of life on Earth. And in fact, what we know now is that life has been mostly multicellular over the whole history of life on Earth. And it was not until recently that we started to see multicellular life and then plants, animals, and so on. And we primates are really here at the very tip of the evolution of life on Earth. And one of the implications of this is that this microbial species has been evolving over very long periods of time, all sorts of mechanisms of interaction between microbes, microbial communities, and also another implication is that the first forms of multicellular life evolved in a microbial environment. So we can actually think that these multicellular forms of life of these eukaryotes really evolved, establishing very specific and complex interactions with these microbes around them. But, of course, among all the microbes that are around, I mean, one of the most important for us in terms of health are infectious diseases, because infectious diseases has been a major threat to human health over human history. We have been going through many massive pandemics over the human history, and infectious diseases continue to be a major threat to human health today, and it may be also a major threat to human health in the near future. By 2050, can be really millions of people could be dying just due to infectious diseases produced by antibiotic resistance, I mean, as a consequence of antibiotic resistance. And so we are going to make another time travel to only 92 years ago, when Fleming discovered antibiotics. And this was also a revolutionary moment in human history, because actually he discovered one possibility, one way that we could deal with infectious diseases, we could treat infectious diseases. But the problem that happened really after is that after the discovery of antibiotics, we started to see right after this that microbes starting to, well, resistance to antibiotics started to emerge really very soon after. So essentially, during the years, the 1960s, we started to use antibiotics in a very abusive way. And then really after, we started to see an increase in the antibiotic resistance until now, around the years 2000, we started to see that antibiotic resistance was becoming a really major issue on infectious diseases, and infectious diseases came back to be a major threat and major problem to human health. So a paper published in 2011, they used an ancient DNA approach to try to understand how old was antibiotic resistance in human populations. Not in human populations, how old was antibiotic resistance as a mechanism in life. And they analyzed a sample for 30,000 years ago, in the moment in which we had mammoths and different extinct species living around. And actually, what they concluded was antibiotic resistance was largely predating the use of antibiotics, something that we could think that is something obvious. I mean, considering that these microbes have been evolving and interacting with each other for very long periods of time. So when I was in my postdoc, something that we thought about was trying to analyze many different metagenomes from ancient samples. And the idea was to try to explore a little bit further the diversity of antibiotic-resistant genes in samples that predated the use of antibiotics, in order to have a sort of sense of the diversity of these mechanisms in environments that were not really affected by the use of antibiotics in modern humans. And of course, what we found was that all the different families of antibiotic-resistant genes were present in ancient metagenomes. But one of the most, one of the interesting results that we got was that we were able to assemble de novo full genes of antibiotic-resistant, different antibiotic-resistant families. But one of the things that we observed is that many of these full genes that we recovered by assembling de novo these ancient metagenomic datasets were having a very low similarity at the protein level with known genes, known antibiotic-resistant genes. So some of them, they showed that less than 70% of amino acid similarity to the best plus hit on NCBI. So one of the questions that we asked was, well, are these genes actually antibiotic-resistant genes or are something else? So we focus on a group of beta-lactamases because beta-lactamases as antibiotic-resistant genes are interesting because they act directly on the antibiotic. So it's much easier to test the direct activity of antibiotic-resistant activity. And actually what we saw by analyzing many ancient metagenomes were that the diversity of beta-lactamases that we were able to retrieve from this ancient sample was much larger than the ones that are known, which are here shown in colors. So one of the questions was, well, all this diversity that we see here that are quite divergent from the known beta-lactamases, are they functional or not? So what we did was to pick up one of the most divergent that we had in our data set. And so it was one that was only 30% similar at the amino acid level to the best hit in the antibiotic-resistant database, and only 45% similar at the amino acid level to the best hit in NCBI. So the full context where this gene was found was classified as Bacillus species. We did a de novo artificial synthesis of this gene. We clone it, we express it in E. coli, and we then did an experimental characterization. So in a way, we were playing to Jurassic Park, try to revive ancient genes and try to see their functions. And what we found was actually that this gene was functional. So here, what we show here is that, so this is a culture of E. coli. When the gene was not expressed, the E. coli couldn't grow next where we had the antibiotic. And when we had the beta-lactamase gene, the E. coli couldn't grow. We tried this with different beta-lactams, and what we found was the phenotype was intermediate between two different families of beta-lactamases, these two over here. So based on the similarity of this beta-lactamase to all known beta-lactamases, on the phylogenetic positioning of this beta-lactamase, and on the phenotype of this beta-lactamase, we concluded that this was really a new family of beta-lactamases. But one of the implications of this discovery is that we could actually start thinking about the possibility of doing a paleo-microbial park. What I mean with this is that we can actually now use these kind of approaches to revive genes and enzymes from ancient microbes and try to see how are they functional. So this was just a proof of concept, but something that can be done at a very much larger extent on any microbe that we can find in an ancient sample, and some of which may be extinct. Right, so now I'm inviting you to continue our time travel much further back in time. So, well, this was a picture that was already shown before in Matthias' talk. And so one of the things that interests me is that, well, humans went out of Africa around, let's say, well, 100,000 years ago. And when they went out of Africa, they started to go to all sorts of different environments, all sorts of different, they started to get in contact with all sorts of different animals, all sorts of different microbes as well. So the question that has interested me for a long time is how was the interplay of these human groups that were moving around the world with the microbes that they were encountering through this journey. And in principle, in particular, I'm interested to understand when, where, and how we got our infectious diseases in this journey across the globe. So it turns out that, I mean, archaeological samples are great windows to the genetic past, and mainly teeth are in particular very interesting because teeth can conserve DNA from humans, of course, because they are highly irrigated tissues. Also from pathogens if they were circulating in the blood at the time of death, and also from oral microbiota and oral microbes. So from a single teeth, we can get the 3 layers of information, and we can do all sorts of studies with this information. In the past few years, high-throughput sequencing has been applied for ancient DNA retrieved from ancient teeth. And actually, different work have been able to recover the genome from ancient pathogens, from pathogens that are from the bacteria, virus, and eukaryotic kingdoms. And so now I'm going to explain a little bit about one of the work that I've done in the past few years. So I will invite you to travel again with me back on time around 5,000 years ago, where we find here in Sweden the first Neolithic farming populations. And in fact, around 5,000 years ago, we have this case of a woman that died together with other people in their village. So there was a work published by Matthias Jacobson a few years ago, actually 2 works of them, where they analyzed the remains of these individuals that died around 5,000 years ago, and they published two highly interesting papers about the history of these early farming populations in Sweden. But what I did was to reanalyze that data and try to see where we could find traces of why they all died in this period of time. And what I was able to recover was 80% of the genome of Yersinia pestis, which is a bacteria that causes plague. It was not only the genome from this bacterium, but also the genome from the 3 plasmids that are typically found in Yersinia pestis. In fact, there was not 1, but 2 individuals that were infected with Yersinia pestis, and it turned out that these 2 individuals were older than the oldest cases known at that point of plague in Europe. So at that point, the model that was that we had about the presence of plague in Eurasia was that plague has come to Europe by the steppe migrations that took place from Eurasia to Europe around 5,000, 4,000 years ago. The problem with this model, or with the result or my finding comparing to this model, is that we were already finding a plague in Europe before the steppe migrations. So we had to find another explanation of why plague was found in Europe at that moment in time. So essentially, the scenario was such that we were finding plague at very distant places in Eurasia, very far in the east, over here and over there in Europe, in a rather very short period of time. When we analyzed the genome of these Yersinia pestis strains, it turned out to be basal to all known species of Yersinia pestis to date, including those that were moving around Eurasia in the Bronze Age. And when we analyzed the genome in more detail, we found that many of the SNPs that were accumulated in this genome found in Sweden were only found in this particular strain and not in any other Yersinia pestis strain. All these are Yersinia pestis strains. On the other hand, when we analyzed the SNPs that were accumulated in the strains from the Eurasian or the Bronze Age lineages, we found that many of these mutations were shared between the strains found in Eurasia during the Neolithic and Bronze Age, but none of them were found in the COCM2 lineage that were found in Sweden. So essentially what we had is a scenario in which we had one lineage of Yersinia pestis here in Sweden and another lineage that expanded across Eurasia right after. So the question was, where was this lineage found in Sweden coming from? So the next analysis we did was a molecular clock analysis to see how these early branches of Yersinia pestis were diverging. And what we found was all the major lineages of Yersinia pestis were all diverging in a short period of time between 5.7 and 5.1 thousand years ago. So in a matter of only 600 years, we had all the major lineages of Yersinia pestis diverging from each other. So the question that we asked was, what was going on in Eurasia during that period of time that could explain this major or this large divergence or radiation of Yersinia pestis lineages? So essentially what we had in that period of time in Eurasia is that, well, agriculture was adopted in the Near East between 12 and 9,000 years ago. And right after, we see an expansion of all the early farming population from the Near East into Eurasia and Europe. This is still predating the radiation of Yersinia pestis as I just showed. Then what we have is a period between 7,000 and 6,000 years ago of technological innovations where we have metallurgy, wheel transport, and animal traction being adopted in Eurasia. And right after, we see the first trade routes traced over very long distances in Eurasia. And actually, these trade routes start to overlap with the period in which we see the radiation of Yersinia pestis lineages. So for the first time, what we have is human interactions over very long distances. Another thing that was happening in that period of time is that we see for the first time the first mega settlements to ever exist in Europe. So between 6.1 and 5,000 years ago, we have the first human mega settlements in this region here. And actually, the existence of these mega settlements overlaps quite well with this period of time in which we see the plague radiation. And this is for the first time that we find between 10 and 20,000 people living together in Europe. So we can assume that in these settlements, the hygiene conditions were probably not that good. Also, what we have is people living in close association with cattle and animals, so we can assume that they were in close contact with zoonotic sources, and also that they were burned out. We see that these sites were burned out multiple times to finally collapse around 5,000 years ago. Another thing that we find in this period of time, between 5.5 and 5,000 years ago, was that, that there was a large spread of animal traction and wheel transport. So we don't know exactly where in Eurasia this technology was adopted, but what we know is that in this period of time, mostly all populations in Eurasia were using it. So essentially what we have is a scenario in which we have the perfect means for the emergence of an infectious disease such as plague, and for the large-scale spread of a highly contagious infectious disease. So by integrating all this data, what we built was a model of how we believe that plague could have emerged in human populations, proposing that plague could have emerged in the first mega settlements to ever exist in Europe, and then rapidly expanded across Eurasia thanks to these technological innovations I just mentioned. This model doesn't preclude the possibility that of the second model that was proposed by Andrés Baltuenia, that later in time we see that the expansion of the steppe migrations into Europe could have brought some lineages of Yersinia pestis into Europe, and a little further in time back from Europe to the Eurasian steppe. But something that we see really after the emergence and large-scale spread of Yersinia pestis in Eurasia is that the populations in Central Europe declined. So some of them disappeared, some of them reduced largely in numbers, and what we see right after is that there was a massive migration from the steppes into Europe, which caused a completely genetic turnover of European populations. So one of the questions that we have now is that, well, whether pestis and plague could have played a role in the decline of Neolithic populations in Europe. So just to summarize the results that I just presented, in this work what we had is a number of findings. We found the oldest and most basal case of plague in Sweden. We found that plague was already in Europe before the steppe migrations from the Eurasian steppe to Europe. We found that there was a large-scale radiation of basal Yersinia pestis lineages between 6,000 and 5,000 years ago, and also that massive human migrations did not initially spread the pathogen. But for me, what is also interesting about this kind of works is that it can also propose new hypotheses that can favor further work. So in this case, the hypothesis that we propose is that pestis could have emerged in the first human megasettlements to ever exist in Europe, that plague could have contributed to the decline of Neolithic populations in Europe. And given that for the first time we find in the continent the perfect conditions for the emergence and large-scale spread of a pathogen, that this could have been the first pandemic of human history, which is, of course, is just an hypothesis. So essentially, our findings contributed valuable information about what we know about plague, but I hope that it will also inspire and be used to drive future research. So just to conclude, the next stop of my journey will be the southern cone of the Americas, and this will be the main focus of my lab in Institut Pasteur. And the idea would be to analyze Native American populations, ancient remains from Native American populations by integrating the three levels that I mentioned before: the human genetics, the microbial genetics, and the whole microbiome information. So with this, I would like to thank you, and I would like to also particularly thank Simon Rasmussen from Copenhagen, that we did together the work on Neolithic plague, and also the two anthropologists that participated in the work that is presented about pestis, because without them it would have been impossible to build and to integrate all the layers of information that help us to build the model that I proposed, I presented. So thank you very much, and happy to take any question.